Not moving slides. Sorry, these main services you can find also on web pages. But if you do not find what you like, you can every time you can contact us and we'll discuss, because we do also some other type of analysis, for example, cosmetic analysis, characterization of products, organic synthesis, negative barriers, so DNA impact analysis. So we can discuss, and if we will not violate our experimental setups, maybe we can try also something a little bit different than we usually do. So that's I wanted to say. We work predominantly full service mode. It means you will bring samples and we will give you the so you do not participate in this protein analysis at all. Report you will get all details on analysis, and we are able also provide you with some statistics and some basic bioinformatic evaluation of your data. So it depends what is the aim of the experiment actually. And you can reach us on our own booking system, which also serves as a communication channel. So we prefer this communication to email communication, because emails usually are lost after some time. But in our system, everything is archived. And we look back and so even if you can get us online, you can order what you want. I would prefer if we can talk prior each proteomic experiment to design also experimental setup in your lab to optimize the whole experimental design to get the best results as we can. For example, to remove possible batch effects or contaminants which interfere with our mass spec analysis and so on. So if you plan some more complex proteomics experiment, just contact us before you start. That's all. Our core facility is quite has a quite large community of users. In last year, we have almost 200 users, and out of them, about 40 students. So, if you look on distribution of community, most of users comes from our mother institution, from CPEC MU, and from the other parts of MU, mainly Faculty of Science, Faculty of Medicine, and also we have some others from Faculty of Pharmacy. But we have also quite a portion of academic users from Czech institutions. Also. If we look on composition of actually instrumental demands to our core facility, if we do it based on number of orders, most of orders goes to MOLI-MS and second to LC-MS. But if you look for instrumental time which you spend on your samples, LC-MS is of course prevailing, because we have several LC-MS systems. All these LC-MS orders are much more demanding on time. So it's just a brief overview. As I said, you can approach us using our booking system. Unfortunately, the unifying with central booking system will not be done actually, and you can approach us directly on this web page, but you can also use our web page which is SAPEC. Concerning pricing, because it is also the most important part of our work. The most important change is, as was mentioned also in previous talk of Hauser, the CISB discount will be reduced to 85% instead of 90%, which is now, and we will pay from next year 15% of our full costs. Here, concerning data, here I would like to mention that we still are able to archive all raw data and also process data at least for 5 years. And so we don't, because we are working on full service, we don't normally automatically don't provide raw data to users, but of course we can do it on request, usually by file sender, and it's not a problem. And what we provide, if you publish data of proteomic analysis, now it's standard that the data should be deposited in some public repositories, and we can do it for you. Usually we use Bright database. It's no problem. Just contact us and we can arrange. Of course, we can provide textbook methods and such kind of things. This is just regarding instrumentation. Our instrumentation that didn't change from last year. So we have already mentioned multi-LS instrument. And we have 2 timsTOF instruments and 2 orbiter-based instruments. And as I said, Exploris and timsTOF Pro instruments we are using for this whole proteome analysis for the differential proteomics purposes, mainly, and also for analysis of post-translation modifications. And all these instruments are connected to C separation. So those 2 instruments in our lab. Sure. Still we have some queue. So this slide is from the field of might happen. Again, it's dependent on CISB investment call. And here we would like to obtain another timsTOF instrument, this time Ultra. This is the last generation of this timsTOF technology, which provides ultimate sensitivity, and it's directing to so-called single-cell proteomics. I would say we would be able to analyze low input samples, which I can demonstrate here on companies results. If you look, for example, for this part, this is analysis of 30 picograms of protein digest, which correlates with, for example, this neutrophil, this protein content of neutrophil cell. And we can identify, at least the company could identify almost 4,000 of proteins and quantify about 3,000 of proteins, which comes to be interesting because now we could see quite a portion of proteome, at least at this basic level of protein, identified proteins. And here we could really approach this intercellular variability, or we can distinguish differences between small populations of cells in different tissues, not only in human cells but also in plant cells. So it would be really interesting if we could get this instrument and we could learn efficiently use it. So that's something for the future. And at the end of my presentation, I would like to mention some some of our outcomes. So last year we had 37 acknowledgements, which was like a large involvement, our core facility in different kinds of studies. And I would here like to thank you for acknowledging us. You heard it several times, and I again repeat, it's critical for us. We need acknowledgements if our work is mentioned in some publication. And I selected one example quite recently of publication where we did some proteomics. It's a work of Professor Lukeš from České Budějovice and Professor Jurčenko from Ostravská univerzita, University in Ostrava, sorry. So it's work on some such kind of organism, and it's an example that Mother Nature makes exceptions, and they prove that this organism codes in different ways. So usually codons which are stop codons now are they possible they code in this case glutamic acid or tryptophan here. So and here in this study we provide evidence for this at the protein level. So we did proteomics on it, and also it's an example of wrong acknowledgement actually. So please do not acknowledge people from core facility. We need the number of projects. Yeah, so they apologize I exposed, but yeah. So as I said, this is our acknowledgement text, and we also usually add another acknowledgement because we quite heavily use computational resources of Institution of Computer Science here. So we also ask for acknowledgement for this project. That's all from my side, and so thanks for your attention, and I wish you sun in your minds.
Thank you, Zbigniew. And since we still don't have any questions online, so there is enough space for you to ask Zbigniew. Now, maybe 1-2 questions.
Maybe if I can ask regarding this option to deposit the raw data in the repository. So it works in such a way that upon request from the user, you do everything, you just provide them with the final code, or?
Yes. Yeah. Usually at first this reverse code because it's not public. So yeah, we just to discuss which data we should deposit, and that's all.
And how huge volumes of data do you have by?
Very rare, about 50 tera.
I want to ask issue, and if you also work on some protein modifications like phosphorylation and analysis, for example, phosphoproteomics.
Yeah, it was at the second slide actually. Phosphorylation is our main modification which we are working on, because they are quite interesting from biological point of view, but of course from methodological point of view it's also very easy. So it's most frequently studied applications, and you can come and you can do. Okay.
You know, Zbigniew, I just checked the acknowledgement of the Nature paper, and this is the example where the grant number is not mentioned, and only mentioned the names.
That's what I just.
Maybe I didn't pay attention to what you said. I was just searching.
I will mention it in the end, actually. So I asked people not to acknowledge people from core facility. It's not correct.
You know, they did even some correction. they published to this paper,
Yeah,
there is a addendum, so they could have modified also the funding.
I suppose you know you are.
Yeah. But it doesn't excuse him.
As I said, they apologized by email, but